Hello, everyone. Welcome once again to this week's CAP seminar. Today we have with us Muammar Mansor. He's a research group leader at the geomicrobiology at the University of Tübingen in Germany, and he has received a PhD in geosciences and astrobiology in 2017 from the Pennsylvania State University in the United States. This was followed by a postdoctoral fellowship in environmental nanoscience at the University of Texas El Paso in the United States as well. And he has carried out various interdisciplinary projects, which are generally centered on biomineral formation by microorganisms and their impact in understanding past biosignatures and the modern fate of various nutrients, elemental cycles, toxic metals in waters like carbon, phosphorus, nickel, and antimony, and also the future of sustainable and technological materials in Earth. So thank you very much for your presence here, and go ahead.
Okay, so thank you very much, Miguel, for the introduction. So as you said, my name is Muammar Mansor. I'm really happy to be here to present at the Centro de Astrobiología. And so actually for my first slide, I would like just to introduce myself a little bit more because I was thinking about my connection to astrobiology. And so this is a picture of myself about 10, 12, 13 years ago. And so I was a biologist. I was doing a Bachelor of Science in biotechnology, and there was a lot of emphasis on application and the usage of microbes in order to develop useful products. But I knew back then that I had a lot more interest than just that. And so I knew that I had a lot of interest in earth science and also in astronomy. I was taking a minor in astronomy. And then one day I was just walking down a hallway. This was in Penn State University in the USA. And I found somebody working within astrobiology. And I was really interested because astrobiology is something that is really interdisciplinary. It combines all these different fields, and I could learn so much more than if I just stayed in biotechnology. And so fast forward a couple of years, and then I did my PhD. I continued at Penn State University by combining geomicrobiology and astrobiology. I was under the tutelage of Professor Jen McLady. She is a very avid caver. She likes to explore life in the subsurface, in the dark, in extreme environments. And one of the type of life that she found was this thing called snortites. This we typically found in very sulfidic cave. And what's interesting is that this drop of snot essentially has a pH of less than 2. And when we look at them under the microscope, they are extremely, extremely rich in bacteria. One specific type of bacteria in particular called acidithiobacillus. And so this is life that is based on sulfur oxidation. They actually produce sulfuric acid that contributes to the acidic pH. And they also perform dark CO2 fixation. They are actually the primary producer in the deepest part of the cave. And then fast forward a few years. So I stayed in the US for a couple of years. Then I went to Germany about 5 years ago. And I expanded my research a little bit more. And now I'm really working on how microbes make minerals, how microbes dissolve minerals, and how that might have implications to biosignatures, fate of nutrients and toxic metals, and also how these biogenic minerals maybe can have application for future sustainable materials. And for this talk, so I'm going to focus, of course, on microbial life. Why? Well, because they are the simplest life forms in the universe. So I think we all are very familiar with this, that early life on Earth, in the first 3.5 billion years or so, they were almost exclusively microbial dominated. And today we can find microbes or extremophiles living in so many different environments, from the deserts of the Death Valley in California to the depth of the ocean, to acidic and sulfuric hot springs. And by understanding these different environments, it then allows us to understand the limits of life and how we can apply this to different astrobiological sites, other potential habitable worlds, including Mars, icy moons, and also maybe other moons, other types of moons that might exist in our solar system and the universe. And so my talk today is also about biosignatures. And in 2022, we wrote a review about the different types of biosignatures that one can look for in the environment. And here's a summary. We have 5 different types. The first one that's really easy, that kind of people can relate immediately. So if you see something that looks like a cell, This could be a cellular microfossils. They might also produce different rock fabrics and textures. They might produce minerals that have unique morphologies that might be attributed to life. We can also look into geochemical signatures, whether in the form of organic matter or in the form of trace elements and stable isotopes. And today I'm just going to focus on two of them, being that microfossils and mineral precipitates, specifically more focusing on their morphology, their shape, how we can use them in order to detect biosignatures. And for this, it's really, really nice if you work with iron oxidizing bacteria or iron microbes. Why? For example, we can find them in different environments. And so this is a video that I took from the southern part of Spain in Rio Tinto. So we have these biofilms, this really slimy, stringy material that we can find forming today in the river. And over time, they become lithified, they become solid, they become something that we can hold with our hand. And these kind of deposits, they are all iron oxide deposits, rust essentially. And we can find them, not only in Spain, but across the world. This is another amazing example where we found this 1 meter long iron stalactites in the Black Forest in Germany. And all of these deposits, we know that they are formed by modern iron oxidizing bacteria. We have iron 2, they are oxidized by the microbes, and they form these deposits called biogenic iron oxyhydroxides. Sometimes we call them bios, just to shorten them up a little bit. And so this orange structure, these are all bios. And these microbes can produce these iron deposits using oxygen. And when they produce it with oxygen, we found these amazing microstructures. For example, there are some microbes that can produce these twisted stalks, and some microbes that can produce these sheath or tube-like structures where the bacteria then resides in the sheath. And they produce these organic templates simply because they want to protect themselves from encrustation. So you imagine that you're a bacteria and you have iron minerals forming in solution, and then suddenly all these iron minerals want to attach to you. They slow you down. They block nutrients from going in and out of the membrane. So bacteria have some certain mechanisms in order to protect themselves against encrustation. But even more than that, so these are all aerobic metabolism. We also have bacteria that can perform this iron oxidation without any oxygen. For example, we have those that can use light energy in order to oxidize iron. We also have those that can use nitrate. And here they couple it to the fixation of CO2. And we also have those that cannot couple this to carbon dioxide fixation. And in that sense, we need to give them a source of organic carbon. And these are representative image of these three types of bacteria. And what you can see is that they have clear differences in the degree of their cell-mineral association. The phototrophic and the nitrate reducer tends to be quite separated from the minerals, while those of this mixotrophic nitrate reducer, the one that requires organic carbon, they can be fully encrusted by these iron minerals, which means that some of these deposits may have potential as biosignatures. But what I'm really interested in, especially something that I've worked on for the last 3 or 4 years, is how these biosignatures can be destroyed, essentially, or how can they be transformed. And we know that when they get formed in the environment, then they need to go through a series of processes before we can see them in the rock record or in whatever environment that we are interested in. So if you go down and look at the different microbial respiration processes that exist in the environment, you can predict, for example, that at the top you have aerobic respiration, and then there are microbes that uses nitrate, manganese, and then once you deplete all of this respiration process, then you get to the zone of iron reduction. And so these iron oxide deposits can be transformed in this zone. If you go down even further, then you get to the zone of sulfate reduction, which means that you can produce hydrogen sulfide. And hydrogen sulfide is another reactive component that can react with iron minerals. And then you go down, you get to the zone of methanogenesis. And if you go down even further, then you get to the zone where no microbes can live, but you get even higher temperature and even higher pressure. And then you get to the different metamorphic zones. And all of these processes can destroy carbon, and they can transform iron minerals biosignatures. And so for the focus of the talk today is that I'm going to talk about the long-term fate and preservation of microbial iron deposits. It's divided into 3 parts. In the first part, we're going to focus on this iron reduction zone, looking at microfossil preservation within iron minerals. And then we're going to look into going deeper into the sulfidic condition, how this transformation can transform the minerals to make biogenic pyrite. And then we're going to go down even deeper to higher temperature, looking at how certain microfossils can be preserved under this condition. And so jumping right ahead into part 1, microfossil preservation within iron minerals. And in some slides, I'm going to show the picture of the person who's responsible, who's heading for the project. For example, here we have Laura. She's a PhD student of mine, and this is her project. And she, or we, I should say, are very interested in how all of these different biogenic iron oxyhydroxides can be used as food, essentially, for iron-reducing bacteria. This is a scanning electron microscopy of the iron-reducing bacteria Geobacter sulfur reducens. And you see this kind of ridges on their surfaces. This is very distinctive of Geobacter. And they can reduce iron 3 to iron 2. And they can, at the same time, they also degrade organic carbon to carbon dioxide. And so in our experiments, we simply make these bios, and then we feed them to the Geobacter sulfur reducens, and then we see what happened. And when we did that, is that when the first experiment that we did not add any extra organic carbon, but we can track the amount of iron 2 that is produced relative to the total in the system. Then we tracked this over 50 days, and we saw that only about 50% of the iron 3 can be reduced, which means that the iron has been transformed. And if we add acetate, which is a really easily degradable organic carbon, then we see actually that within 10 days, about 100% of the iron 3 can be reduced. So these bios can be easily transformed by microbial iron reduction. And the extent of the transformation depends on the availability of organic carbon in the environment. But what's more interesting is that when we then look at the transformation products under scanning electron microscopy, and so we had these 3 different bios, and for the first 2, more or less, we see the same thing. We saw the formation of vivianite, which is an iron 2 phosphate mineral. They have this characteristic blade-like tabular structure. And we saw a lot of cells on the vivianite, mostly those with this characteristic ridges that we think are Geobacter. But what's more interesting is that for this third type of bios, where there's encrustation, we see evidences that there might be actually cells in vivianite. We see more fractures, we see more holes. And this is not the only picture that we have. We have more under different conditions, under different time points. We see that, okay, so here, for example, you see one cell that might be poking out right here, and here as well. Here you have a cell that is almost fully covered by vivianite, and here as well. So we see this kind of microfossil preservation, essentially, where the cells are being fully entombed within this vivianite. But when we saw this, we were like, oh, okay, that's great, that's amazing, but we were also very skeptical, because we know that when we prepare the samples for scanning electron microscopy, we had to do a lot of steps in between, centrifugation steps, and this might cause some artificial artifacts in doing the imaging. And so could this be just an artifact of sample preparation? And are the cells fully entombed or not? Or are they just like partially covered? And also we wanted to know which cells specifically. Is it the iron-reducing bacteria, or was it the previously encrusted iron-oxidizing bacteria? And so in order to answer this question, we contacted our resident expert in scanning electron microscopy, and she was able then to perform focused ion beam milling. And so you're going to see this video where we have this. So this is a piece of vivianite, and she then uses this focused ion beam in order to cut away layer by layer, revealing what's inside the vivianite. And so here you can see that as we get down into a certain stage, we see that there are actually cells that are actually quite fully entombed within the vivianite. And that's great. That answers one of our questions, but we had more. So for example, we wanted to know, then we also tried a different technique. This is called the cryo in situ fluorescence coupled to X-ray mapping. This is something that that we are developing together with Cristina Escudero, my collaborator. And we went to the diamond light source in order to do high resolution imaging. What's cool about this technique is that you can do cryo imaging, which means minimal sample preparation. We can couple this to fluorescence in situ hybridization. Then we can tell apart which ones are the iron reducing bacteria, which ones are the iron oxidizing bacteria. And then we can do 3D mapping for X-rays element-specific carbon and iron. And so here you see this is one piece of vivianite, and this is one piece of vivianite. And there seems to be one cell that is partially encrusted with the vivianite. And so we are still going through the data and the images because it's like hundreds of gigabytes of data to process, but more to come for that in the near future, hopefully. And so to summarize part one, we've shown that under iron-reducing conditions, we think that some iron oxidizing bacteria can be entombed within vivianite as microfossils. And since we've done this for a few vivianite crystals, then the size of the vivianite crystals really matters as well. It cannot be something small. It has to be big enough to fully entomb the cells, which means that we can tell apart some of the environmental conditions that this might happen as well. And this is very interesting because vivianite has only recently been identified in Jezero crater on Mars. So if we can get those vivianite nodules back to Earth, and then we apply the same techniques, maybe we can find these microfossils here as well. Okay, so that's part one under iron reducing condition. And then part two, we're going to focus more on biogenic pyrite. And this is the work that is being done by my PhD student, Fatih. And so we've gone through this area where we have different types of iron minerals. Usually for the bios, they're composed of two different types of iron mineral, ferrihydrite and goethite. And if we go down, then they get transformed to vivianite. But if they get buried even deeper, then we reach into the zone of sulfate reduction. And in this zone of sulfate reduction, we have a lot of sulfate-reducing bacteria. They reduce sulfate to form hydrogen sulfide, coupled to the degradation of organic carbon, and then they form various types of iron sulfide minerals. This can be, some of them have really weird names, makinawite, greigite, and then pyrite, I think is the one that's more commonly known. You don't need to know all of them in detail, but the reason that I am mentioning them, bringing them up here, is because they, the names indicate different identity, and then that indicates different sizes, reactivity, and also their preservation potential. Essentially, when you have something small, like ferrihydrite, these are less stable in environment. They can transform over time to something like goethite, which has this larger crystal, more crystallinity and higher and larger structure. And so they can be preserved much longer in geological timescales. It's the same thing when you get into this iron sulfur minerals, iron sulfide minerals. Makinawite has this crinkly structure, but they're very small. Greigite is just a tiny bit bigger. They're also magnetic, which is interesting. And then pyrite can be nanometer to something centimeter that you can hold and see with your hand. And pyrite in particular is very, very important for many reasons. The oldest pyrite have been found at about 3.8 billion years ago, which means that if you then analyze trace metals, isotopic composition, morphology of the pyrite, it may tell you something about the record of the seawater and biological activities. Pyrite are also forming actively today. It is estimated that about 200,000 tons per day is formed on Earth. They can have a range of sizes, going from very small to very big. And they also have very interesting morphologies. You see some of these pyrite, they are euhedral, cubic structure, but then some of them have these rhomboidal structures where they are composed of microcrystals that aggregate together. And when you look at them under the microscope, it's really amazing to look at them because they're shiny, they are easily distinguished. And when people noticed this the first time, actually there was an idea going back to Paul Ramdor, who was a German scientist back in the 1950s. He suggested that when he saw these rhamboids, he suggested that they were fossilized colonies of sulfur bacteria. Nowadays, we know that it's not so simple because you can roughly easily make abiotic rhamboids as long as you know the recipe. And the recipe is just that you mix iron, you mix sulfur at high concentration, and then you heat them up. Typically, you require temperatures over 80 degrees Celsius, which means that this may not be fossilized colonies of sulfur bacteria. But so that doesn't mean that microbes are not involved in their formation at all. What we want to know is that can microbes really form pyrite rhamboids or not, especially at ambient temperatures, because this has not been shown experimentally. And one of the challenges is that we know that it's difficult to form biogenic pyrite in cultures, let alone rhamboids in microbial cultures. I did this during my postdoc where it was a complete experimental failure. We had sulfate-reducing bacteria, and I give them carbon, I give them iron, and then they form these black minerals. And these black minerals, then I let them sit there for about 2 years, and there was just no pyrite forming, and it stopped at this intermediate species of Machina white and greigite. And so that was like a really failure in the experiment. But then looking at the literature and trying to piece together what people know, actually it's not so easy. You don't just need sulfate reducers and iron to form pyrite. You need more than that. It's much more complicated because actually you need all of these different processes. You need iron-3 minerals that can be reduced by iron-3 reducing bacteria. You need all these different sulfur intermediate species. And then following the combination of all these processes, then you can form pyrite. And this is really challenging to replicate in the laboratory because you need all these different microbial species, intermediate species, and you need to track all of them. So what we did is that we came up with a simplified pathway in order to form pyrite. And so we then use the same bacteria that can reduce iron, Geobacter sulfur reducens. As the name implies, they can also reduce elemental sulfur, and they produce hydrogen sulfide. And so we can delete, essentially, some of these pathways. We can simplify them. And then we have Geobacter that can produce both iron and hydrogen sulfide. And so we can do this somewhat easily in the laboratory. Okay, so this is our idea. This is our drawing board. Let's try it. So we have ferrihydrite as a source of iron, sulfur. We try an iron-sulfur ratio of 1:2 at circumneutral pH. And you can see that in the control, we have this brown coloration, which is typical of iron-3 mineral. And then after 2 months, we have this black mineral precipitates. So then we analyze this using Morse power spectroscopy, which is a way to identify the mineralogy, and no pyrite. After 2 months or so, we're still stuck at Machina white, greigite, this intermediate iron-sulfur species, iron-sulfur minerals. And also, actually, interestingly, we formed some vivianite. Okay, so that didn't work, but we did get some cool SEM images because here you see, okay, these are the Geobacter, and then these globules, these are elemental sulfur. And after about 15 days or so, they get coated by this wrinkly structure, which we then determined to be Machina white. So it seems like there's this local microenvironment where the minerals tend to precipitate. Okay, that's okay. Then we can do another attempt, the third attempt. And as it turns out, we found out that iron-3 to the sulfur ratios matter a lot. We tried conditions where there's a lot of iron. We tried 1:2, which is stoichiometrically for pyrite is iron-1, sulfur-2. And then we tried 1:4, which is much higher sulfur. And then we found pyrite. So at day 50, we observed that there's already something weird going on. We can use a magnet and we can say, okay, this is when we detected greigite, which is magnetic. Then we use a technique called nitric acid extractable iron. So most of the iron minerals that I've mentioned, they all dissolve in hydrochloric acid. And so we can dissolve them, we can remove them, And then what's remaining are pyrite that's only dissolvable in nitric acid. And so we quantify that. And so we can see that within the first 2 months, already there's some pyrite forming. But within 6 months, it's very clear. Pyrite formation is about 70% of the total iron in the system. And then we confirm this with X-ray diffraction. Within the first 15 days, we have machina white, we have vivianite, iron phosphate. And then within 4 to 6 months, then we have greigite and pyrite, only pyrite at the end. So great, then we can form pyrite. And just going to take a look at the vivianite just for a short while, because vivianite, so it has these crystalline beladed minerals, which are very typical of this crystal. And we can track its formation and dissolution by using geochemical analysis. This is a really nice figure that Fatih prepared. Essentially, here you see that in the first stage, you have iron being released in the solution as a lot of iron reduction is happening. And you also see phosphate decreasing. And then you have vivianite being formed within the first 30 days or so. And then after that, you get a rise in sulfide and you get a release of phosphate in the solution. And this is actually where vivianite is then dissolving. At the same time, these gray bars here track pyrite formation. So you see that pyrite formation only really increases or only really starts when you have free sulfide in solution. So we can track vivianite formation, dissolution, but also when the iron sulfide mineral starts to increase and starts to form. And how does the pyrite look like? Well, after 4 to 6 months, we see a lot of these structures. These pyrite are very, very small. They're about 1 micrometer in size, and they all look more or less like spheres that can be surrounded by very small crystals, maybe machina white, and some organic matter. We don't see any cell incrustation, and we don't observe any femboidal pyrite after 4 to 6 months. Okay, that's good. But we can play around with something even more. So we know that in the environment, there can exist different types of iron minerals. And we tried 5 different types that we synthesize in the laboratory. We can try very, very small ferrihydrite, goethite that can have 2 different sizes. We can try magnetite, and we can even try the encrusted cell. And by doing all of this, we show that, okay, we have a recipe that we can form pyrite at ambient temperatures under all conditions, no matter what type of iron minerals that we use. But what's interesting is that they all produce pyrite at different rates and at different extent. One thing that I want to highlight here is that using the encrusted cells, the BIOS, we actually see quite low pyrite formation. And we think that some organic matter is actually inhibiting pyrite formation, but this is still being studied. And so we can come up with a general sequence of pyrite formation for all conditions. We have concurrent reduction of the iron minerals and sulfur by geobacter. This then allows for iron 2 to be adsorbed on sulfur particles. And then we can form machina white specifically on the sulfur particles. And then this age over time, this creates a zone where there's a lot of reactions happening. And then we start to see pyrite also starting to encrust the sulfur particles. And then over time, what we see is that the sulfur particles disappear. And sometimes you see these structures where there's a shell of pyrite remaining. And so we can form pyrite clusters, but not femboids. So these pyrite clusters, so here are some images where you can see better. So this is a really, really large pyrite cluster. This is a typical shape of an elemental sulfur particle, but they have been fully replaced with smaller size pyrite. And this is the shell that I've shown you before. And I just want to highlight also a couple of observations from these experiments. Using BIOS as the initial material, we saw that there are some cells with this wish-like structure. We think that these are geobacter and they are not encrusted. And at the same time, we see this cell right here, which has this encrustation by something weakly, most likely machina white. And this seems to be like an iron sulfide encrusted cells of iron oxidizing bacteria. And in one setup in particular, the one with nanogoethite, We see again pyrite that has a lot of these ferrule to maybe euhedral particles. But look at the pictures in the bottom row here. We see crystals that seem to aggregate together into more or less a spherical shape. And this roughly fits the definition of femboids. We're not really sure if these are real femboids. So at the moment, we call this either femboid-like or just mini femboids. And so that was really exciting when we were able to see this. But there is a problem because if you compare that to something that we typically see in the environment, they are extremely, extremely small. In the experiments, we see something that form a maximum size of 1 micrometer. In the sediments, it goes to 5 to over 50 micrometer. And if you put this to scale, actually, this is the size of the femboids or mini femboids that we are forming. It's so small. And so that's the conclusion for part 2 for the moment. We have shown that under sulfidic conditions, iron minerals transform to mixtures of iron sulfides and pyrite. And we can form different pyrite morphology, but no clear preference for femboids. So at the moment, our conclusion is that femboidal morphology is not a biosignature. But this brings more questions. Do we need more time for them to grow to larger sizes? Or maybe we need to use different microbial species. And another thing to consider is that we did form vivianite, but they disappear under sulfidic conditions. And this can have implications to part 1 of the talk, in which you can have microfossils preserved with the vivianite, but under sulfidic conditions, they get destroyed. Okay. So now moving on to the last part of the talk. So this is the work by my PhD student, Hervoye, in collaboration with Jan-Peter Duda from the University of Göttingen. And we wanted to look at how filamentous microfossils can be preserved in the rock record. Why? Well, one of the starts to this project was really when we look at this paper that came out in 2024. This is a picture that they published. And what you see here are these things from 518 million-year-old deposits that they suggested to be microfossils. You see this filamentous structure, and then you see that in individual cells, they are actually completely filled with pyrite as well. And so there's some amazing images in there. But what they suggested, they go even a step further. They suggested that these are actually fossils of sulfate-reducing bacteria. And I was a bit skeptical when I saw this because I wasn't sure on how exactly this can happen. And we also see a lot of similar structures in hydrothermal deposits, hydrothermal vents. We see these organic laminations that are fully coated with pyrite. And could this be fossilized filamentous bacteria? And so we wanted to test this in the lab. We used this approach called experimental diagenesis or experimental fossilization. We had some questions whether the filamentous mats and associated minerals can be preserved under sulfidic conditions at high temperature. And can specific structures or textures be used as biosignatures? And to do that, we need to first collect the samples. So we need to go to environments where we can find the samples. And so this is one stream in Switzerland. It's a sulfidic spring. It's very cold. It's in the Alps. But here you can see that we can collect lots and lots of filamentous microbial mats that you can just scoop up with your hand. When you look at them under the microscopy, you see these clear filamentous structures, and you see inside that they have these bright structures. These are all elemental sulfur inclusions. Then we also have this field site in the mines about 2 hours away from our location. And you see these iron deposits just fully covered the mine. The puddles in the mine as well as on the walls. And looking at the microscope, then you see these twisted stalks and sheath, which are typical for iron oxidizing bacteria. So we collect them, we brought them into the lab, and then we put them under different conditions. This is a typical experimental fossilization setups. Here, starting from the sulfur mats, we add a source of iron because we know that when they get buried, they will get exposed to iron. We mix them together to observe how they attach to the mats. And then we add sulfide, which then causes the reaction to go really, really rapidly. Iron minerals transform to iron sulfide minerals. And within minutes, you see everything turn black. And then we put this at high temperature, incubate them at 80 degrees Celsius for 2 weeks. And we use a variety of techniques in order to identify their mineralogy and to look at the cell-mineral association using SEM, using X-ray diffraction, and using sequential extraction. And so starting with the sulfur filaments, when we did this to the sulfur filaments, what we see is that the filamentous structures are in general preserved. They can have this encrustation of the minerals, but what's interesting is that they are not encrusted by iron sulfur minerals. They are encrusted only by elemental sulfur. And we think actually that this elemental sulfur is coming from inside the cells, being released and then precipitating outside to the filaments. But there's no pyrite. And then if we do this with the iron filaments, we get completely different results, actually. What we see is that, so this is how the filaments look like in the beginning, and the filamentous structures are preserved, and they get completely covered with the spheral to euhedral pyrite. And these are some of the smallest pyrite particles that I have seen in the environment and also in the lab. And so to summarize, under sulfidic and high temperature conditions, we see that filaments of sulfur oxidizing bacteria can be preserved and are coated with extracellular elemental sulfur. And there may be a chance for the sulfur then to be replaced by pyrite, but we have not seen this in our experiments. In comparison, filaments of iron oxidizing bacteria are well preserved and fully coated with pyrite grains. And so the biosignature potential for them is quite high. And we are then continuing the experiments to look at preservation at even higher temperature and pressure. We have ongoing experiments up to 250 degrees Celsius and 1,400 bar. Okay, and then just to summarize what we've seen or what we've learned from this talk is that in part 1, we've seen that microfossils can be preserved within vivianite under iron-reducing condition. Then under sulfidic conditions, we have transformation of the iron minerals to non-framboidal pyrite. And at the same time, we saw that vivianite can be dissolved and the microfossils can be lost. And then in part 3, we saw that the iron oxidizing filaments can be preserved well, but not necessarily the sulfur oxidizing filaments. And this is related to pyrite encrustation. And so, of course, this is implications to if we consider how we look for biosignatures in different environments. And with that, I will then end my talk. I want to thank the geomicrobiology group, especially Andreas Kappler, who has supported my research, Stefan Fischer and the Tübingen Structural Microscopy core facility. All the electron microscopy images come from there. They're amazing. Yeah, and I'm happy to take any questions.
Thank you so much, Muammar. This was an incredible talk. I will have a few questions later, but now we have a pretty decent time for questions, so.
Thank you very much. I like very much your talk. I wonder, all these fossilization experiments, how complete are they? I mean, how far is the incrustation process? Does this process replace everything in the intracellular content or not? Mm-hmm. Can you explain a little bit more? Because I don't know, you haven't saw any experiment about the molecular content of this, the carbon and all the biochemical stuff that perhaps is still there. Yep. But so can you explain a little bit more of that? Because somehow I could imagine that this encasement of the yep. cells is somehow to make a very isolated environment that might help to preserve the organic matter inside. Yep. Can you explain a little bit more about this?
Yeah. So really great question. So I can answer this from several different angles. So for example, for the iron oxidizing bacteria, where we have this cell encrustation, we see that the age of the cell matters a lot. At the beginning, you see only some partial encrustation. And then if you let them age over time, then you get full encrustation. And then you can actually see that the iron oxide starts to go inside the cells. So the whole cells can be fully encrusted in a sense. But we also know that we only see this at high iron concentration when we supply them with a lot of iron. Now, in our lab experiments, we provide millimolar levels of iron. But in the environment, we only have micromolars or so. So it is still a big question for me, even, that can we see these encrusted cells or not? In terms of the twisted stalks and sheath, those for sure, we can see them in the environment. And I think what we haven't done is we haven't put the twisted stalks and the sheath under iron-reducing conditions and then see whether they can get fully covered by the vivianite. I think that will be very interesting. Now, in terms of like going even smaller and to see like, okay, what parts of the organics is actually preserved within these minerals. So we haven't done that. And that's something we haven't touched. Certainly something that we're keen to do that in the future. Maybe something to do with carbon zanes or something. But yeah, if you have ideas, I'm also happy to hear them.
Any other questions?
Thank you. I've enjoyed so much your presentation. I have one question regarding, you've quoted the recent article by Horowitz from Jezero Crater about the potential consideration of some patterns on top of a rock that they were called leopard or poppy seeds as a potential biomarker. So your experiments are run in 3D in a fluid, right?
Yes,
Everything grows three-dimensional. But I guess when we look at the surface patterns, we need to have something that is grown on a surface. Can you guide us how to interpret what happens when that is being grown on a rock, and why would such a ring pattern be produced?
So short answer is no, but I can provide some of my ideas at least. So mineral formation that happens on surfaces versus mineral formation that happens in suspension or solution, they occur quite differently, or at least during the initial stages. Once you have this mineral in solution and then you form these minerals, then you have new surfaces from which to grow. But I would assume that for these structures that we see on Mars, it might be that we need to start with something that is more similar to that. So we need a surface. And then from there, then we put in the liquid suspension because we always need water. But we can also do something that's like semi-dry conditions. And then we see how the vivianite might grow under those conditions. So that's the way that I would approach this experimentally. But as far as I know, these patterns, leopard patterns and so on, these are also quite unique and not something that we've seen on Earth so far, on vivianite. Yeah. That's a great idea. Yeah. I might steal it.
Hello, Muammar. Great talk. I enjoyed it a lot. Beautiful talk. I have two questions. The first one is related to the possible biosignatures of many of the minerals that you have shown. Vivianite, sulfides, for example, pyrite, or white. So you have talked basically about structural, microstructural features. Do you have, or do you know of any chemical radiochemical signature that can be related with microbial processes or that could be used in addition to the structural features?
So this is definitely something to explore in the near future. I would say in terms of sulfur isotopes, in terms of iron isotopes, I'm not so convinced because the fractionation during mineral precipitation itself can be mixed with abiotic processes and biological processes. So it's really hard sometimes to tell them apart, at least in laboratory cultures. But when it comes to trace metals, I think this is really promising. So the different rates of formation, whether it's abiotic or biological processes could be higher. And then you also get contribution from organic matter that might be trapped within the pyrite particles. And then you might have different amounts of nickels, or cobalts, and copper, and so on. And I think this is really, really cool to explore in the near future. The issue or some technical challenges is that the pyrite that we are forming is very, very small. And so we need something that can really like distinguish all these different mineral species very, very clearly, maybe using some sort of sequential extraction method. At the same time, we need microscopy method that can really map all this at maybe even nanoscale. So there are some technical challenges, but I think this will be great. And also, actually, if you're interested, there is one group in Paris who's looking into this in more detail, but from the abiotic side.
Okay. But you haven't identified a trend in the trace metal so far that can be okay.
No,
That's
not
what
yet.
I thought. The second question, and last question, is related to the framboidal pyrite.
Yes.
Yes.
I knew it.
You know, yeah, you know, it's probably one of the minerals that has been studied so more extensively in history, especially framboidal. So you have obviously and it's nicely shown that there's no need for microbes of microbes to form framboidal pyrite.
Yep.
You have shown a very nice model of framboidal pyrite. But my question is, even though you know of a mechanism that can produce a microbial mechanism that can also produce framboidal pyrite, and in this case, which chemical features, which chemical conditions, physicochemical conditions, do you think that would favor the formation of framboidal pyrite in relation to microbes?
Okay.
If you think it's a possibility.
I think it's a possibility, but this is a very difficult question. So I can imagine two mechanisms, and both of them are related to organic matters or biofilms that the microbes produce. One is that we've shown, or we have some suggestions here, that organic matter can actually inhibit pyrite formation, which means that the minerals, as you build up iron and sulfide, you actually increase the supersaturation of the minerals. And so when that happens, you encourage nucleation, new nucleation of pyrite grains compared to growth of existing pyrite minerals. And so you can form these tiny, tiny crystals much easier when you have this organic matter inhibition, as long as iron and sulfur keep increasing. So that's one mechanism that I can think of. Another mechanism is that you need a way for the framboids to assemble together. And this, I think, can be encouraged if you have pores or porous structures within biofilms. Essentially, the minerals, when they form, they cannot form randomly. They need space in order to form. And so if you can form these minerals within pores, and then they have no choice but to assemble into a sphere. So those are the two mechanisms that I can think of. Yep.
Thank you, Muammar.
Any more questions? Okay, I have a couple mostly methodological ones about the geobacter experiments.
Yep.
So at some point in one of those experiments, you were like, yeah, this is clearly geobacter. This is more like an iron reducer bacteria. So what exactly were the inocula that you used there? So it was not just like geobacter culture. It had other bacteria or...
No, so it's only geobacter.
Okay.
Pure geobacter.
Okay.
Yes.
Then I misunderstood that.
Yep.
And also in another one experiment a bit before, where you added the acetate and then you saw that the iron 2 went up. What exactly you added in like the non-acetate culture, because they need to feed off of something, or do you just added them without any source of carbon and see what happened?
Yes. So what's interesting about geobacter is really great. It's a model organism. And actually we can control how much they reduce iron versus how much they reduce sulfur by changing the pH. And this is all controlled by the thermodynamics of the energy that they can gain from all these different metabolisms. And with geobacter, we know that they can usually degrade simple organic carbon, which means like something like acetate, good. But we were also interested, well, we have this model organism in the lab, can they degrade the organic part of the biogenic iron oxide hydroxides? Can they degrade complex organic matter? Can they degrade the cells completely? So we wanted to test this, and this is why at first we did not add any acetate. And then we see, okay, well, only some of it can be degraded. Then, okay, now we add more organic carbon. That's something that they like. And then we show, well, technically, as long as they have enough organic carbon, they can reduce all of the iron tree.
Thank you. Are there any questions in Zoom or no? Anyone else? Yeah. Then I think this is all for today. Thank you so much.
Thank you. It's my pleasure.